Thank you everyone for coming here and hear about our project proposal. It's called SymBioWatch, from symbiotic biology, and watch, like watching, monitoring, but also watching because it's a wearable. And as I said, our technology starts from a state-of-the-art technology that's out there, partially validated. The minimally invasive technologies, such as devices to monitor biological events for diabetic patients, for example, are out there. Now we want to capitalize from that and push to another level in a long-term goal, which aims for something that is super important and critical for us. The problem is we get sick, and age is a major driver for most of the age-related diseases. The problem is we don't know really how to handle ourselves. We're all different. And if we could prevent, know in advance what kind of lifestyle, treatments, or medicine we can take, we could prevent most of, or at least postpone in time, the onset of the age-related diseases in general, and increase our health span. That's our problem we want to solve. And our vision is that we could solve this problem by collecting information in real time and learning, accumulating data, also in a collective effort by many, many people, in reaching critical mass that will be able to predict in a personalized way how we could prevent the development of degenerations that leads eventually to disease. So we're talking about a market that is pretty much every single living person, if you want to stay healthy. That's a big vision. Now, to be practical, we want to start with one simple indication, which is already out there, and implement a technology and a market and a patient population that already exists and already uses these kind of devices. So make me a favor now, look on your left and on your right. Include yourself, and at least one of you are going to have diabetes if you live in the United States. That's what data suggest. So that's actually the very first population that we want to target. Now, our aspirational second indication is breast cancer, cancer in general. Of course, early detection of these patients to prevent the development of cancer is critical, and there is a body of knowledge of biomarkers that we could use now that could be implemented in our platform. Starting with breast cancer, which is the major cancer population, without saying the major market in the oncospace. Of course, our long-term vision is longevity. And that cannot be done easily and is a long-term goal. However, we said by building a platform that is user-friendly and collecting data, pretty much like in a model of 23andMe, you start with a simple idea, but as you keep collecting data, at some point, computing it, and we can build a predicting algorithm that can be, in a personalized way, giving you feedback that can help you to keep track with your health state. So state-of-the-art, technology that wants to use blood and to kind of give information to predict your health state are out there. Even examples like a failure like Theranos, in reality, was extremely successful. Why? Because it's an extremely powerful idea. So we want to do something that actually works. There are technologies out there that already include FDA-approved system that can be integrated and measure biological events. And even like major companies such as Wearable, Google Life Science, ex-former Google Life Science is working on that, developing systems such as BioWatch, which there are a number of patents out there. Okay, what do we do? And we want to start from scratch, so I presented really hand-scratch model of what we're proposing to do. So we want to integrate several systems in developing a minimally invasive device. So how we envision is literally having like diabetes patients do, a microcannulation that collects blood samples and from the system, but like is connected to using flexible biomaterials, connected to a wearable device, such as a watch. That is a micro laboratory. And our idea is the system can collect cells from bloodstream or proteins for extracellular matrix and sort the cells and analyze, lyse the cells, and enrich the sample using a atomically based build nanopore membrane, and eventually analyze that. And using a microfluidic system and microtechnologies, nanotechnologies that can actually all be incorporated and has been shown in literature, patented or published. So our main technology is based on plasmonic detection, and pretty much using a micro analysis of light emitting through the device. All the parts are absolutely implemented already in micro devices. You can be used a conductive ink, like a gold ink, into microfluidic device in that. Our team has all experience in academic and industrial field. We are currently running this kind of work in academia, or we previously built, like myself, a company who's currently developing this technology in microfluidics. And I'll tell you a little bit later what that shows. Now, by implementing this for detection of proteins, this has been shown. So people use this technology to detect with high sensitivity single proteins, combining antibodies that are conjugated with the heavy metals. So having a high specificity, running this through the system, and you can detect it. Now, this system in microfluidics has been shown to increase by 10 to the 10th, 10 to the 11th time the sensitivity of normal benchtop devices based on spectrography. So back to our system. The simple workflow is literally, we have 2 data points collection. Number 1, from the blood can we collect cells? And I'll show how, but it can be implemented using microelectrophoresis, capillary electrophoresis, and again, antibodies isolating cells from blood. And this is really the first point that we can use to collect data. So we can quantify cell types in the blood. After lysing in the microfluidic lab on chip, there is this protein enrichment through the nanopore. This is to increase the sensitivity of the system and remove the background noise. But this can also come straight from the extracellular matrix and directly enrich the protein, again, using antibodies label. And we have ideas of how to overcome the antibodies and use other technologies to do that. And I'll tell you in a second, but enrich the proteins and sort them again, collect data. So there will be 2 levels of data collection, and we have ideas how to implement more, like adding, for example, DNA, RNA analysis into that. As I said, capillary electrophoresis is something that has been demonstrated in many, many ways and miniaturized, is highly sensitive, and can be built on board, on chip, using MEMS and other micro technologies. Similarly, in Raman spectroscopy used for the plasmonic detection, laser has been replaced with LED as an excitation source, easily implemented in microchips. And we expect to build our first prototype in about 6 months. It will be a relatively large device as a proof of principle. Again, combining technologies that are out there, expertise that we have, connected and collaborated with companies we are working on or we also co-founded before. So we have all the expertise to do that. And we already know the biomarkers for our first indications to pick which proteins we want to use, both in diabetes, which is step 1, breast cancer, also started as a proof of concept. There's a huge need to develop that, because even in early detection of of diabetes, pretty much pre-diabetes is undetectable currently, unless you have a constant monitoring and more expensive and invasive ways to do that, just by keep collecting blood over time. So our milestones, we built to, number one, we aim to build a large prototype first and develop a computational platform. Our team is a stellar team with this expertise done already in several companies before. Together then we want to miniaturize that and debugging the different steps. So we believe we can build the different parts, combining, miniaturizing it, and work on increasing the detection of more proteins. Eventually use this to validate a signature that can be also improved as we enrich our database in the cloud. That's the idea that the more users, first a niche of populations that need and have to use that and already use similar technologies, expanding to other patients' population. Eventually, everybody might want to use something like that. The more we collect data, the more signatures can be done. And even protein that can be used, that can be, updated in your device by changing the cartridge with the reagents. So criteria for evaluating our progress. First, we want to see proof of principle. Can we made a prototype? Can we detect early events for diabetes or in cancer? Again, going through all the points. Can we do assembly the parts, validation sensitivities, building the computation platform, integrate with machine learning, and most importantly, test with human blood samples to validate our model, and test in animal models as a proof of principle for the in vivo implantation. Now, for bio device, the FDA track can be super fast. A device like that has histories with Red FDA to go in this validation up to 6 months. It's not like a drug. So technology transfer, we believe we can build value in doing that. We can certainly capitalize from the plasma resonance amplification signal, integrating what we're doing and build better technology by miniaturizing it and integrating it with, in our platform. We want to also go beyond antibodies. And by building libraries of DNA-based aptamers, cheap to do, stable, can be used at room temperatures, design them, screen for them, and replace antibodies with aptamers. So something that is already like an ongoing state-of-the-art technologies. And of course, the nanopore-based technology we want to optimize. And as a second step in the aspirational work, do that also using nanopore to do DNA sequencing, which is already up there and is already commercially available in the size of a USB drive. Funding sources for that, we expect to reach out to venture capitals, starting with angels and SBR grants and foundations. We expect to build the first prototype in 6 months, latest. We can do that in a shorter time with our starting team. That's expected budget. We think you can achieve that in 2 million, including budget and people and material, going quickly through the seed and aim for a Series A in between 6 and 12 months, raising minimum of 10 million for the next phase and miniaturization and putting together the work. The team, we believe we already have most of the expertise required from bioengineering, aging, longevity, regenerative medicine, the biomaterial, nanomaterial, nanopore development, machine learning, all the patenting, all the work in biology related to what we're doing in aging, and the microelectronics and microfabrication. So we're ready to build a research proposal, move forward, SBI grant, reach out to angels. After that will be incorporation and build a prototype. We can actually do that very quickly. Long-term payoff. So the vision is in 20 years, we see how these people, everybody wants to use a device like that, such as a wearable right now. And as we do that, we can build a large database, as I said, with an increasing predicting power. We want to integrate other technologies as 2.0, 3.0, where we integrate also DNA, RNA, and reach out a full implantable miniaturized device, maintaining a minimally invasive approach. The idea is to go to a health state, not necessarily a pathogenic state, monitoring people, reaching out a signature that can prevent the decline correlated with age, and give advice in a personalized medicine approach that includes nutrition, sleep, exercise, medications, and is fully integrated with the healthcare system. So the idea of an aggregation of this data from millions of wearers should allow us to really reach out a next stage of personalized medicine as we really envision. And with that, I'm very happy to take any question you might have. Thank you.
Questions from our judges?
Hi. So the reason we have continuous glucose monitors and not continuous protein monitors is what I understand that there's a much higher concentration of glucose in blood. And current technology for immunofluorescence measurement of protein concentrations would require more of the vial of blood to get that blood test. Do we know that plasmonic detection has a much higher sensitivity than standard imaging techniques?
Yes, we do. And the increasing sensitivity is 10 to the 10th, 10 to the 11th times more than the current state, which we believe we can even increase by integrating in a microfluidic device, which is what we are actually already doing. So the answer is yes. Then they can also minimize the reagents, which is also minimizing the cost, and increase the sensitivity by working with these micro scale volumes. The other problem is that glucose is not a good prediction of pre-diabetes, and the only way is to use protein detection. So we believe we can create a signature, a predictive signature that of course needs to be validated, but this already is, the literature is out there, that cannot be done just by monitoring glucose.
I absolutely agree that real-time protein quantification would have lots and lots of useful applications.
Yeah, so where's the atomic precision?
In the nanopore membrane. This is like a self-assembling on atomic base structure, and that is a critical step. We believe that without that, this system is going to fail because the noise of all the reagents that are going to mix together are going to be too high. So we need to have a way to filter and enrich the protein of interest, and by building this nanopore structure is a key. Is really part of what we believe is a key technology, and that requires atomic precision to be built.
So if when you get to the point where you're multiplexing lots of different proteins, is each protein an individually customized hole in the pool or in the membrane?
Yeah, they can mix and match, but again, after lysation and mixed through the serpentine in the device, this solution can be split in multiple channels. Each channel is designed with a membrane to detect each individual protein. So you're going to have an array of, initially it can be just 3, 10, 20, 50. So you just lyse, mix, divide, and analyze in each microchannel, which are different. But you can also potentially mix and match the same nanopore membrane.
Can you zoom in on the sampling of the plasma? How do you get some amount of it? Is there anything nano there as well?
In the what? Sorry?
The sampling of the blood. How do you zoom in on it and give us a little more?
After lysation,
Getting it from the blood.
the all steps. Or how do you get from the blood?
Yeah, getting the blood into the beginning of your
Okay.
diet.
Yeah, it's a microcannulation. It's pretty much like microcapillaries that the same way are currently used in certain medical device. Is minimally invasive, but it requires to be inserted into a vein. And it will collect, it doesn't have to be constant, but it will collect at set time a minimal amount of blood. They will suck in and will be processed into the system. There is the cartridge that we envision is a small, that includes the waste. So we don't know how often this could be done, but that's kind of part of the disposable of the system where there are the reagents, including like the aptamers, the buffers, and the waste. So when you remove that and you place the new cartridge, you have the new reagents or different reagents potentially to detect different proteins, but you also remove the waste that you collected.
Anything else from our judges? Okay, I think we are out of time, so thank you.
Thank you.